Hello, good morning. I'm a little bit nervous because we are moving to a more macro view about these polyploids. So be patient with me. I'm feeling like an architect in a congress of civil engineers. It's a little bit scared. So my idea, of course, is to get a different perspective, and I would like to share with you something about where these beautiful alleles are inserted and how is the behavior of these structures during the evolution. And in the final, I hope you can get more informations, or you can get new parameters for your model, right? Okay. So, of course, before we start, I would like to thank all of the team involved in my laboratory. So we are in Brazil at Exalc, and this is an honor to be here with you. So today I'd like to talk about this work that is in progress, revisiting the Polyploids in legumes. And then I will divide this presentation in 4 main parts, starting with the motivations. What about Polyploids in legumes? Analyzing some Polyploids species, and then make some final remarks. Right, so about the motivations for this work is because we have amazing genome comparative analysis in different models, like in cereals. This very classical work made by Gale and Davis, it's pretty amazing, helping us a lot about the chromosome structure and how these chromosomes are behaving during the evolution, and of course how the genes are distributed along the chromosomes. And also we have these same ideas and analysis made for brassicas, and this is a wonderful work made by Martin Lizac, reconstructing the history of the chromosomes in this species. And also we have something made in legumes, but a long time ago. And the idea in my group is to revisit some of these structures and then to bring new ideas about the chromosome organization and the evolution. So the other motivation is because the landscape about the Polyploid is quite complex. So for me as a cytogeneticist, and after many years working with the chromosomes, I have this synthesis about Polyploid. So for me, I have a real autopolyploid when I am inducing the Polyploid in a specific species. Or I have a real halopolyploid when I am inducing the Polyploid in a hybrid between different species. So, and why I have these ideas is because after you produce your polyploid, immediately this material starts a process called diploidization. And then you have this evolutionary process that create a range of segmental polyploids. So all of our polyploids are in different stages with different structures, with different genetic behaviors. This is the motivations, right? And then what about the Polyploids, the Polyploid in legumes? So if you look at the phylogeny, most of the species we have been analyzing, they are real diploids. But in some points, we have some species with the double of the chromosomes, or sometimes more than the double of the chromosomes than most of the species. And then for these conferences specifically, I would like to share with you some of these 4 cases, and mainly analyzing the most distant species phylogenetically in relation to Medicago truncatula, alfalfa species that has a very nice assemble of its genome. So let's go to analyze some of these Polyploids species. So firstly, Glycinimax with 4 chromosomes. What we did was a dot plot comparing the Medicago genome with the Glycinimax genome, and then we reconstruct the chromosomes based in the piece of the Medicago genome. And then in Glycinimax, we could get very nice results, as you can see here in green. This means that the homology is very good. And then we can see that the Medicago genome is spread in all of the chromosomes of Glycinimax. And then the idea is to make some reconstruction to infer about the polyploidy in Glycine, because this is a paleopolyploid. We don't have too much information, especially in the cytological level, to infer precisely, for example, the level of the polyploidy, because this I have this interrogation here. But as you can see here, I pick up just one example. The chromosome number 8 is split in 4 different chromosomes. So this kind of information can be helpful for us to understand the origin of this chromosome number and the structure for soybean. Arachizipodia, peanuts. So peanuts is well known as a halotetraploid, and then we know the parents, the parents here, Arachis duranensis and Arachipaiensis, and we did the same thing. So this is a real halotetraploid that we can know exactly from where it is coming, and then we make this. We made this reconstruction too. And in this case, we had some problems, as you can see here in red. This means that the homology is not very good, and probably we had some problems in the sequences. Okay, but it was possible to reconstruct some idea of the chromosomes. Despite we know the origin of the polyploid, we don't have that theoretical organization of the chromosomes. Some chromosomes appear as a single chromosome, not or without a homologous chromosome. But even though we have in some points some homologous that can share some homologies, and also they can be rearranged during the evolution. Only in one case we found full homologous chromosomes. So, and finally, taking some more distant legumes in this genus Lupinus, they have a very smaller genome than Medicago. We also tried to make this reconstruction using the dot plots and making some draws of the chromosomes again to get an idea. And one of the points for us is to understand we can keep some blocks of the chromosomes across the evolution. And then, surprisingly, even this group with 4 chromosomes, we could find some interesting results, like this group of chromosomes. They have some kinds of homeology, and the structure are very similar. I keep the chromosomes in the original position just to give you an idea. Probably when we refine these analyses, we can move the chromosomes. Probably this is not real inversions, just because the molecule is not in the right orientation. But even so, we could find, for example, here, these 4 chromosomes. That means, well, probably, maybe this is a tetraploid species in its origin. So, and also, again, we can find some homologous chromosomes, and maybe useful for information. So the other Lupinus species has 50 chromosomes. Of course, we follow the same approach. And then we could find only homologous chromosomes in double, like here, here, and here. But because of the homology with Medicago truncatula, we are not completely sure about tetraploid origin of these species, but we remain analyzing these genomes. So it's very quick, no? Just 15 minutes. Well, I'm feeling like Usain Bolt. So this work is in progress, but we believe that legumes has a proper model for the evolution of the karyotypes, different from brassicas and cereals. This is very important for us because we download the sequences from the databases to make this kind of analysis, and the quality of the assembles is really important for us, and we are finding some problems if the genome is not well assembled. Of course, we cannot make good inferences about the chromosome structure, and especially in a evolutionary perspective. So another point, back into here, we are using the whole genome, or it means I am including the repetitive DNA in this first analysis, and of course would be interesting to mask the sequences and use only the position of the genes to make the same comparison. I hope to do this soon. The other point is we hope this kind of information may be helpful for breeding programs and of course for genetic mapping. Also, you have a different perspective, and of course we have a mix of behaviors if you consider your alleles inside one species. Also, we hope to develop some probes to make fish in very distant species to check if these informations get from computer analysis is true, and we can confirm this in the samples using the chromosomes. And finally, the polyploid is a very complex topic in legumes, still being, and its analysis is an important source of information about genome organization and evolution. And I hope you can get some ideas and other perspectives about how to make models to analyze polyploids in legumes. So, and also, we need collaborations and partnership to go deeper and finish this analysis. So thank you so much.